Welcome to the presentation of our online course, Simulation, Scale-Up, and Optimization of Microbial Fermentation. Through our online courses series, you learn the principles of bioprocess engineering and their applications to enzymatic bioconversions, microbial fermentations, and animal cell cultures processes. For more details, visit us at bioprocessengineeringonline.com. In this course, you will explore the methodology for the digital scale-up and optimization of fermentation processes. You will learn how to design and utilize simulation models or digital twins of microbial fermentation to predict and optimize the operation of industrial fermenters. The methodology will be taught through a detailed case study on glutamic acid fermentation. The goal is to optimize the industrial scale use of a thermo-inducible strain of Corynebacterium glutamicum, which excretes high quantities of glutamate in response to a temperature increase. The course is divided into two parts, each consisting of 4 sessions. Part 1 focuses on the kinetics and modeling of the fermentation at the laboratory scale. Part 2 covers the scale-up and optimal operation of the industrial fermenter. Each part can be taken independently. In Session 1, you will learn how to perform kinetic analysis of bacterial growth in both batch and continuous fermentations. You determine the specific rates of cell growth and sugar consumption. Additionally, you will identify the kinetic laws that describe how these rates depend on the composition of the fermentation medium. In Session 2, you will determine the kinetic law for the specific oxygen consumption rate. You will also analyze the limiting rate of oxygen transfer from the air to the medium and calculate the characteristic oxygen transfer coefficient KLA. In Session 3, you will analyze the bacterial metabolic kinetics in response to a temperature increase from 33 to 39 degrees. You will determine the specific rates of glutamate excretion and of the simultaneous excretion of the co-metabolite trehalose. Furthermore, you will identify the kinetic laws that describe how these rates depend on the composition of the medium. In Session 4, to conclude the first part of the course, you will construct the simulation model for the temperature-triggered glutamic acid fermentation. Using the Balances thermodynamic kinetic triangle approach, you will establish model relationships that combine the kinetic laws governing cell growth and metabolism, the laws of oxygen solubility and oxygen transfer, and the mass balance equations that describe the time variations of substrates, cell, and metabolites concentrations. In Session 5, to begin the second part of the course, you will scale up the fermentation simulation model to the industrial 200 cubic meter fermenter. The model will be adapted for the larger volume fermenter by accounting for changes in oxygen solubility and transfer rates. Additionally, the model will incorporate potential limitations in the removal of fermentation heat generated inside the fermenter. In Session 5, you will also use the simulation model to optimize the batch fermentation at the industrial scale. Through virtual simulations, you will identify the 3 operational variables: the initial sugar concentration, the temperature increase time, and the final fermentation time that result in the highest annual glutamate production for the batch fermentation process. In Session 6, you will evaluate the production cost of glutamate by considering the entire bioprocess, including both upstream and downstream operations. Using this integrated fermentation and economic simulator, you will optimize the batch fermentation process to achieve the lowest possible glutamate production cost. In Session 7, you will use the simulation model to operate the fermentation in a fed batch mode. After partially filling the fermenter and starting with an initial batch, the sugar feeding will be controlled to maintain a low sugar concentration inside the fermenter. The fed batch fermentation under sugar limitations will be optimized to achieve a significant reduction in the excretion of trehalose and the lowest possible glutamate production cost. In the final session 8 of the course, you will intensify the glutamate production of the fermentor by using oxygen-enriched air. To overcome the oxygen transfer limitations in the fermentor, you will implement a dissolved oxygen control system. Using the fermentation simulator, you will evaluate the potential increase in cell concentration and annual glutamate production, resulting from this additional oxygen regulation. To support your learning, the course provides detailed guides explaining the bioprocess engineering methodology, including the underlying concepts and mathematical relationships. You will also use specially designed spreadsheet templates for your evaluations, featuring a green-red correction system that provides immediate feedback on the accuracy of your inputs. You can progress through the spreadsheet templates independently with the help of a detailed guide, or choose to collaborate with your tutor for personalized guidance and support through video meetings. You have the flexibility to tailor this online course to your individual pace. Plan and structure the course according to your own schedule. Additionally, coordinate the interactions with your tutor through video meetings. For further information on the course, please visit our Bioprocess Engineering online website or contact us at bioprocessdigital.com.